cell organelle was first described by Robert Brown, and later the material of nucleus stained by the basic dyes was given the name chromatin by Fleming. The interphase nucleus, the nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, and the nuclear matrix and one of the spherical bodies is called nucleoli. The nucleoplasm contains nucleolus and chromatin. The nuclei are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm, as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. In the interphase nucleus, where there is a network of nucleoprotein fibers called chromatin, there are different stages of cell division. The cells show structures called chromosomes in place of the nucleus. The chromatin contains DNA and some basic proteins called histones. Every chromosome essentially has a primary constriction or the centromere, as you can see in this diagram, and on the sides of which there are these structures called the kinetochores. The chromosomes can be classified into four major types. These are telocentric, acrocentric, submetacentric, and metacentric. These are the types of chromosomes. The metacentric chromosome has middle centromere forming two equal arms. As you can see, two equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the center. So there is one short arm and one long arm. So these are called as the submetacentric chromosome, where the centromere is slightly away from the middle of the chromosome. In acrocentric, the chromosome has a centromere situated close to its end, close to one of its end, forming one extremely short and one extremely long arm. And the telocentric chromosome has a terminal centromere. So there is a telocentric with terminal centromere, acrocentric with one short, one long arm, submetacentric slightly away from the center with one short arm, one long arm, and metacentric where both the arms are exactly equal.
You can see by the end of telophase, the nuclear division has completed, and now the cell enters into the next stage, that is cytokinesis, in which the cytoplasm will also divide and the cell will form two daughter cells. Here you can see that the cleavage furrow is not complete but will get completed by the end of cytokinesis. But there is slight difference in the cytokinesis of animal cell and plant cell. In the case of animal cell, you see that the cleavage furrow begins from the periphery of the cell and moves towards the center, and hence when they meet, they divide the cells into two different daughter cells. While in the case of plant cells, the cell starts dividing from the center and it forms a cell plate. This cell plate grows towards the periphery of the cell and ultimately it will meet the end and divide the two cells equally. Now when the cytokinesis takes place, so in cytokinesis the organelles like mitochondria, endoplasmic reticulum, Golgi complex, and in case of plants, plastids, etc., get distributed equally between the two daughter cells. There are some organisms in which the karyokinesis is not followed by cytokinesis. Example you can take of liquid endosperm of coconut. So in this, cytokinesis does not occur, only karyokinesis occurs, and because of this, it attains syncytium. Syncytium means multinucleate condition. This is also found in some fungi which show multinucleate.
Mitosis or equational division takes place in all vegetative cells. It is divided into 4 stages: prophase, metaphase, anaphase, and telophase. Prophase is the first stage of mitosis. In this phase, the chromatin material untangles and form distinct chromosomes. As you can see on your screen, centrioles also move towards the opposite poles. This phase is followed by metaphase, during which the nuclear envelope disintegrates and the spindle fibers formed from the centrioles attach to the kinetochores of the chromosomes. The chromosomes are pulled towards the center of the cell or the equatorial plane. Let's move on to the next stage, which is anaphase. In this stage, the centromeres split, and as a result, the chromosomes are split into 2 sister chromatids. Now the chromatids are pulled towards the opposite poles. Let's move on to the final stage, and that is telophase. In telophase, the chromosomes lying at the poles lose their identity and the nuclear envelope reappears. After this stage, the cell prepares itself for cytokinesis or cell division. And what happens at the end of the mitosis? That's right, 2 identical daughter cells are formed.